Hi everyone, let's continue our discussion of the Travisano and Lenski paper, and in this discussion we will try to bring in how history can impact, how can we quantify the relative contribution of history in dictating evolutionary change. So remember that what we have been discussing so far has been in the context of the following, that we had an ancestral trait, ancestral value, then we evolved the population in some environmental context and we have the evolved trait value. And then we have this x equal to y line, and now if this was the ancestral value, if the evolved trait exhibited values above this line but all at the same time, this was the case where there was selection only. On the other hand, I could have a case where the evolved lines exhibit different values but moved from the mean. So, the ancestral value was, trait value was S naught, and in the evolved lines the values are not the same but all of them are above S naught. In this case, the action is evolution is being driven by selection plus drift. On the other hand, if the evolved lines show trait value such as this, which is distributed around S naught, some of the evolved lines exhibit trait value which is above S naught, but there are other evolved lines which exhibit trait values below S naught, then we say that evolution is being driven by there is no selection because the mean is still at S naught and change is being driven by drift only. And we can quantify these numbers by sort of looking at the exact distribution and performing statistics on this. Now, how do we study the effect of history? We make a slight modification. Now, the ancestor that we start with, there will not be one ancestor, but there will be several ancestors. Let us imagine that we had an E. coli and this E. coli was evolved parallelly in different lines, let us say 4 different lines. After an evolution, after a period of evolution in identical conditions in these 4 environments, each one of them will exhibit a different value of this trait that I am looking at. So, if I am looking at cell size, then each one of these 4 evolved lines will exhibit a different trait. These 4 different lines become the 4 ancestral trait values that I am looking at. So, maybe line 4, cells grew up to be really large, this was smaller, smaller and smaller. So, then these 4 become the ancestral values that I am looking at. When I evolve, now when I, so there are 2 rounds of evolution now. The first round of evolution is to generate the different ancestral trait values. So, we do this experiment and there is bound to be spread. So, this is like this manifesting that when I evolve, if this is the manifestation of the evolved lines, then these lines become my ancestor where each one of these has a different trait value, which is what is shown in this figure, that I evolve and I get 4 different values, and these become my 4 ancestors. In the second round of evolution, I evolve this as the ancestor, but I do not evolve once, I evolve this 12 times. So, each of these 12 start from this particular value. Each of these 12 lines start with this value and this value and this value. That is the starting point, and each has 12 different lines. And then after a period of evolution for T1 number of generations, I measure the value associated with the evolved. I measure the trait value associated with the evolved lines. Now, like in the previous case, I could have different scenarios play out. Suppose the 12 lines, so this is ancestor trait value 1, ancestor 2, ancestor 3 and ancestor 4. These can be marked here. This is x equal to y line. So, this is A1, this is A2, A3 and last this is A4. So, what could be the manifestations in this particular case? What could happen is that the 12 lines which were evolved. So, just so that we are clear, so we evolve this and we get this individual, let us call it ancestor 1. So, I take ancestor 1 and I start 12 independent flasks, 12 independent flasks, evolve it in some environment, identical treatment, let us say low glucose for T1 number of generations, and at the end of this T1 number of generations, I measure the trait value in all 12 lines. So, this is the ancestor. So, the ancestor value for all 12 lines was this A1 value, which was ancestor 1's trait value, and maybe the ancestor 1 shows values such as this. The evolved 12 lines show values such as this. Maybe this is the value that 12 lines exhibit. When I do the same experiment now with ancestor 2 as the starting point, I evolve this for T1 number of generations, maybe what I will see is the following. And with ancestor 3, I will see the following. And with 4, something similar. So, now what is this telling us if the response is like this? In each one of these, there is spread. So, it is not like the 12 lines are responding identically. There is not identical response because each of these 12 lines show a different value in the evolved lines. That means this trait is being driven by, there is a role for drift for sure, because if drift was not there, then all the 12 lines which were evolved with the same ancestor would show the same trait value in the evolved stage. So, there is drift. This is also driven by selection because all 12 lines have increased from their ancestor value. A4 was the ancestor value for all of these 12 lines. All 12 of them are higher than A4 now. They are all higher by different amounts, but the point is that they are all higher. So, since all 12 lines are higher than their ancestor, this change is driven by selection also, which is the same idea here, this change is also driven by a sum of drift and selection, same idea here and same idea here. So, individually looking at these 4 groups, evolutionary change is being driven by selection and drift within these each groups. How do we comment on history? History is important because when I start with A1, I can only go up to this range. When I start with A2, my range changes, A3 range changes and A4 my range changes. So, the range is a function of the starting point where I am starting the ancestor from. That is why this evolutionary response, when I compare the 4 groups not individually, but when I collectively look at the 4 groups, I see that the average, let me plot this average here, the average response is going up as a function of the ancestral trait value. So, whatever was the starting point is also dictating what is going to be the end point in this experiment, and hence when I collectively look at this, I say that evolutionary response in this case, is also a function of history. So, in this in the cartoon that is shown, change is being driven by drift, selection as well as history. What would it look like if one of these forces was not acting? This is A1, A2, the 4 ancestor values A1, A2, A3, A4. This is x equal to y line. What if the response was like this, that the first group had evolved line trait values like this, the second group had evolved line trait values like this, the third had these values, and lastly the fourth group So, in this case what we are noticing is that when I look at this group, because they are different from each other, that means this is being driven by drift. Because the ancestral value was this one A1 and the evolved value is actually much higher than A1 and is spread over this range, all 12 lines have moved in a particular direction. That means this change is also driven by selection. But in this particular case, history has no role, because irrespective of where you are starting from, the final response of the population is in the same range. Hence, history has no role. So, the point being, then depending on when we do this experiment and depending on how we see the arrangement of points in these different scenarios, we can not just qualitatively comment on what factor, what evolutionary factor is driving change, we can also quantify these changes as to what is driving this change. Let us do one more and then we will close this discussion of LTE. What is happening in this scenario? So, A1, my 4 ancestor trait values, A4. Now, in this case, the first ancestral values shows a response like this. The second one the third one and the fourth one. So, what do we see here? We see here that in the first group, for instance, the evolved value is centered at A1. Some lines have higher than A1 value in the evolved state, and some of these lines evolved to have a lower than A1 value. The mean, however, remained the same. Hence, in this case, change was being driven by drift, but no selection. The same idea prevails in the other groups also. Let us look at one more, that if we look at the second group, the mean value of this group was A2. Some lines evolved to have the trait value elevated from A2 to something higher than A2, and other lines had this value of the evolved trait reduced from A2 to something lesser than that, but the mean remained at more or less A2. Again, the same conclusion, that this is possible when drift is acting but selection is not acting, and so on and so forth. So, this is a scenario when drift and selection are not working together, but the trait value is only being driven by drift. What about history? We see that history is definitely acting, because if we look at these four means, then this population has mean A1, which was the ancestral trait. This population has mean A2, which was its ancestral trait, and then A3, the third group's ancestral trait, and A4, the final group's A4. So, the responses are centered around the ancestral values that these populations starting from, and since the ancestral values were themselves different, the evolved values are also different. As a result, this response is also dependent on evolutionary history of the populations that are participating. So, this would be a response case where there is drift, no selection, but history is relevant. So, the idea that I am trying to communicate is that suppose we were to do an experiment like this, and these are simple experiments, but through these simple experiments we can answer questions as profound as the relative contribution of history, selection and drift in dictating evolutionary change, and so on and so forth. So, this sort of concludes our discussion of LTEE. We will now discuss a few evolution experiments that I find of interest. I think these are experiments which have thought simple and have led to evolutionary outcomes which I think are counterintuitive, interesting, of value, and I think are intellectually very curious. The first such example is from a paper around 2010, which was titled "The Evolution of Multicellularity." So, when we in the beginning of the course, when we looked at evolution of life on earth, this is today, this is 4.5 billion years ago, we saw that life evolved fairly early. This was prokaryotes, single celled, and they continue to exist throughout evolutionary history, and they continue to exist today. Somewhere along the line, eukaryotes came, individual cells with nucleus now, and they have existed on the earth for about half the time. However, both these were single celled, but somewhere along the line eukaryotes learnt to live collectively. So, this transition from unicellular to multicellular happened at some point. We now know that this transition actually happened several times in the history of life on earth, and there are several reasons for which we think that this transition happened, where eukaryotic, single celled eukaryotic cells learnt to live together to form multicellular organisms, and these multicellular organisms then diversified into all the complex life forms that we see around us. If you see, we are a collection of a few 10 trillion cells. Every complex organism that you see around us, it's an organization of cells where cells have learnt to coexist as an individual rather than living singly. And if you think about it, that's a great transition to have happened. So, this lab evolution experiment asks the question that can we study this particular transition from unicellular to multicellular in lab. And to me, when you ask a question like that, we need to have an experiment in mind where we can test this idea that would this transition happen. Another important point to consider is that when we do evolution in lab, we ask a question like this, we shouldn't ask, it's an interesting question if it's not a task that cell can do in one day. Suppose I put the cells there and I ask that will they divide 4 times, and that will happen in no time. So, that is not an interesting question to ask. If I ask that, oh, if I add one bacteria to LB media, will it divide 5 generations? And of course, it will. So, it is not an interesting question, but it also shouldn't be a question which will never happen. So, can we, if I ask a question, if I grow prokaryotes in lab, will they evolve to eukaryotes? Now, that's an extremely important question, but it probably won't play out in a lab setting, because this event took billions of years, and we know something about why that happened, but probably not going to play out in a lab setting. So, the question has to be just right in terms of not being too trivial, but also not being so complex that it won't play out in a lab in a reasonable frame of mind. So, how do we do this? So, we need a eukaryote to start with, because prokaryotes do not form these multicellular complex organisms such as us. So, we need a eukaryote, and what the authors took here is Saccharomyces cerevisiae, which is a yeast, a model organism about which we know a lot, and several hundreds, thousands of labs around the world use this, and as a result the collective knowledge that we have about the workings of this organism is far more than we know anything about any eukaryotic cell on the planet. So, what is the experiment? So, remember that when we do an evolution experiment, we are actually applying a selection pressure. If we go back to LTEE and understand the selection pressure that was applied there, we allow for growth. So, growth is allowed, and then 1:100 is transferred to next flask, and so on and so forth. So, the idea being, and this process is repeated over and over again, the idea being that what I am asking the population to do in implicitly when I set up an experiment like this is say the following, that if you are an ancestor, you are like this, but if you are a variant that grows fast, then obviously you will divide faster. As a result of that, your chances of making it to the next flask increase. If you divide slower, your chances of making it to the next flask decrease, and as a result, when I set up an experiment like this, who makes it to the last flask? The fast growers. If you have variations introduced in members of the population which make them grow very slow, those variations are going to be weeded out, because only 1:100 go, 99 out of 100 are actually just go extinct from the experiment. So, the selection pressure that I am applying here is for fast growth, and that allows me to make these observations that as time is moving forward, what is the evolutionary change that I am observing in an experiment such as LTEE, that fitness as it is quantified in terms of growth rate increases via trajectory like this. Early on in the course, when we were discussing experimental fitness landscapes, we saw that the first 5 mutations that happened. So, let us say it took this much amount of time for 5 mutations to take place. First 5 mutations that happened in this experiment led to an increase of 35% amount increase in fitness. That is because by the logic of this experiment, I am selecting for fast growers. To select for multicellularity, we have to change the selection pressure such that that only if the cells are multicellular will they make it to the next flask. If you are existing singly as the cell, you will not make it to the next flask. So, the selection has to be on a property which is accomplished better, which is exhibited better by multicellular cells rather than unicellular cells. And the experiment that I will describe now is just a really simple experiment that these authors set up. We take a tube and we grow Cerevisiae in it. So, eukaryotic Saccharomyces cerevisiae is a unicellular eukaryotic cell. It is represented like this, it is also called budding yeast. So, these are Saccharomyces cerevisiae cells present in the population. I allow for growth to happen. So, this is no different from the LTE experiment that we just looked at. So, during this phase of growth, anybody which replicates faster is at an advantage, because it has a greater chance of being transferred from one tube to another. So, the selection here is for fast growth. Now, we saw that fast growing cells are larger cells tend to settle down faster as compared to lighter cells also. So, what this team did was that instead of transferring straight away 1:100, what they did was they introduced an additional step in the process, which was just let it sit, let the tube sit on a bench for 45 minutes. So, this bench was culture in this tube was growing overnight while the tube was shaking. Shaking does not let cells settle down, it also provides aeration which aids growth. But now, before you did the 1:100 to the next tube, you let the tube just sit still on the bench. What is going to happen then is that cells which are heavier are going to settle down and cells which are lighter will keep floating. At the end of this 45-minute period, we sample from the bottom of the tube and sample and transfer 1:100 cells to the next tube. And what is being done here is that in the first phase, when it was only growth taking place, we were selecting for fast growers, but now we are selecting for fast settlers. And the way this experiment panned out, one great way of settling fast is becoming multicellular. Because you are larger, you settle slower, you settle faster, and as a result you have a great chance of being transferred to the next tube. If you are growing really fast, but you never settle, then there is a very small chance that you will be transferred to the next tube. But even if you grow at a reasonable speed, but you sink very fast, then you have a great chance in this experiment to become a fast settler and hence being transitioned to the next tube. We will discuss some more details about this particular experiment and what has happened since this report came out about 15 years ago in the next video.